Hi everybody, welcome to the very last chapter here in the AMP2 semester. This is chapter 29, and as we'll be going through this chapter, you'll see that we're going to leave out a good deal of material that is in your textbook. So please go with the lecture slides when it comes to preparing for your comprehensive final. So here we are looking at a set of events that lead to all those organ systems that we so laboriously explored in AMP1 and the previous part of AMP2. Everything in our own development started with fertilization. We're going to say a few words about it, although in the reproductive system chapter we have seen the production of sperm and the secondary oocytes. They are coming together, will be constituting a new cell, the zygote, and that's where the development of this new individual begins. We'll say a few words about implantation. We're going to have highlights of embryonic development. We are going to have highlights, snapshots of how the placenta develops. We're going to say very, very little about fetal development with regards to pregnancy, gestation. All we're going to say is the time frame, and labor and parturition, delivery is going to be mentioned very, very briefly in this chapter. So a few terms here. So the gestation period or pregnancy normally lasts for about 38 weeks, give or take a week or two. And this is the period from fertilization to the birth or delivery. The before birth period from the fetus or developing entity's point of view is the first 8 weeks, considered embryological development, and we call this developing entity an embryo. And after that, magically we start calling the same entity a fetus, and again we'll look at only highlights of this development in the lab. In the last lab, we have said a few things about the placenta, the fetal structures that are forming inside the uterus, such as the placenta, umbilical cord, the amnion. So we'll see how they are developing here. One theme that I will mention probably a number of times is that by the end of the 8th week, the beginning of all adult organ systems are visible. They are not in their final form, they are not in their final size by any chance, but the first 8 weeks starts the development of all organ system. You can go back in this textbook and explore. In the fetal development, all those organ systems become larger, more elaborate, and more ready to take on the task of maintaining life right after birth. So let's start with fertilization. And here you see the situation. We are in the uterine tube. Here is the secondary oocyte with a lot of the attached cells. As it was moved out of the ovary during ovulation, all these cells from the follicle stuck to the oocyte, and this whole ball of cells is moving along the uterine tube. And by the time we travel through about a third of the uterine tube, sperm makes their way all the way to that portion of the uterine tube. And when sperm and oocyte meet, the sperm cells, the spermatocytes, are programmed to digest their way through this outer layer called the zona pellucida. And several sperms do this at the same time, sometimes hundreds of them. You can see the surrounding sperm. When the first sperm penetrates through the zona pellucida, a sea change, an immediate change happens within the oocyte. So the oocyte will complete meiosis. The nucleus of the sperm will fuse with the nucleus of the oocyte. And most importantly, the zona pellucida changes into a very, very tough tissue, and other sperm can no longer tunnel through the zona pellucida of a fertilized egg. That mechanism, the hardening of the zona pellucida, is there to prevent another 23 chromosomes entering here. Remember, here we have 23 chromosomes from the oocyte, 23 chromosomes coming in with this sperm. Now we achieved the 46 total chromosome numbers. We don't want to add another 23 chromosomes. Chromosomal numbers, whether they are higher or lower than 46, can lead to the activities of the cell going totally haywire. So it is important to keep that chromosome number 46 no more. So the end of that fertilization is a zygote. That's the name of the single cell. We all started out like that in our mother's uterine tube. In some cases, twins are the result of pregnancy, and so the name of these twins, or the class, is the dizygotic or monozygotic, indicates how many zygotes are needed to create these twins. So let's take a look at this incredibly ingenious artistic rendition. So here is the dizygotic twin development. So here's the process of fertilization. In case of dizygotic twins, for some reason, not only one oocyte is ovulated from the ovary, but here's another one ovulated at the same time. This is exceedingly rare. In some incredibly rare cases, there are 3 oocytes, 4 or 5. So twins, triplets, quadruplets, natural twins, triplets, and quadruplets are the result of accidental ovulation of more than one oocyte. So here's the oocyte, and 2 separate sperms, 2 different sperms, will fertilize these 2 oocytes. And so we have 2 zygotes. Here's the name, dizygotic. And these 2 zygotes keep on developing into the embryo. These 2 embryos will implant into the same uterus. They develop side by side, and this happy mom will have a dizygotic set of twins. Dizygotic twins can be different sex, and when you look at them right after birth or even later, they can be easily told apart. Their facial looks are different, just like 2 siblings that were born 5-10 years apart. So 2 separate zygotes from 2 oocytes. How about the monozygotic twins? Now, here the outcome of the monozygotic twin development are twins that are always the same sex, and they always look indistinguishable. Monozygotic twins are the twins where you see mothers putting bracelets on them, different color ribbons in their hair, some way to identify them, because of course they get 2 different names and you don't want to mix them up, right? 2 weeks after birth. So how could this be possible? Totally identical looking twins. The reason for it is that we start with a single zygote. So here's the fertilization, a zygote forms, everything looks fine, regular, a single oocyte was ovulated, fertilization made it into a zygote, but then the developing embryo splits. For some unexplained reason, this developing embryo falls apart into two pieces. The DNA, genetic material, is identical in these two separated pieces of embryo, and now those two embryos develop on their own. They implant at separate places in the endometrium, and they will develop into separate fetuses, of course, because the genetic material is identical. Facial development, stature, usually hair color, eye color, all identical. So these are the identical or monozygotic twins. The next set of slides we'll just consider a developmental process where there's only one zygote present and no twins. So here comes the oocyte. You can see the ovary, the ovulation, the fimbriae will usher this oocyte into the uterine tube. Here's the first third of the uterine tube. You can see the sperm swimming all the way up here. Fertilization occurs right here. And then this embryo, first the zygote, then the embryo, keeps dividing. So cell division, mitosis, preserving that 46 chromosome number, keeps going on. And in about 5 to 7 days after fertilization, a blastocyst forms. And by that time, the cilia and peristalsis of the uterine tube have pushed this developing embryo into the lumen of the uterus. Now, it's really interesting. This artistic rendition brings it out really nice. Look at the size of the oocyte. Look at the size of the blastocysts. They are identical. The cells, as they undergo mitosis, keep getting smaller and smaller and smaller. So the size doesn't increase. There's no ballooning of the embryo here that could block its passage into the uterus. It's really important that the embryo is pushed into the uterus. This is where we see the stratum functionalis of the endometrium. We have talked about that a good deal in the reproductive system chapter. If the embryo is somehow slow moving through the uterine tube, then after about 6-7 days, the embryo becomes so sticky that it will be impossible to move any further, and implantation may occur right here. Implantation in the uterine tube is called tubal pregnancy. Tremendous rate of mortality comes with this because the uterine tube is not equipped, or let's just say ill-equipped, to support embryonic growth as opposed to the stratum functionalis, which is perfectly prepared. So why wouldn't this embryo move all the way into the uterus in 6-7 days? Think about one sexually transmitted disease, chlamydia. Chlamydia, this is a bacterium that lives inside the cell. That bacterium lives inside the cells, epithelial cells of the uterine tube, and scar formation is the outcome of our immune reaction to chlamydia infected areas, and scar formation creates speed bumps here in the uterine tube, physically slowing down the movement and blocking the movement of the embryo. So this is the danger of chlamydia, that it can cause infertility because of this very slow passage of the embryo through the uterine tube. Another way and a reason why this movement can slow down, smoking can slow down the movement of cilia. So one of these moving factors, the cilia, can be slowed down by smoking, and smoking is also associated with the too slow movement of the embryo into the uterus. So a few more words about the blastocysts. So before the blastocysts, there's a stage called morula, and that is a compact, totally solid ball of cells. With the blastocysts, this is a new feature that a liquid-filled cavity develops, and the cells divide into two groups. On a upcoming slide, this will be a lot easier. So this is our blastocyst. This is what is going to do the implantation of sinking into the stratum functionalis. I'm going to move on one more. So here's the best visual on this blastocyst. This is what it looks like when you cut it open. Here is one cell layer, the trophoblast. And here's the other cell clump, the inner cell mass. The trophoblast cells now have a more defined identity. These are the cells that will build the placenta. These are the cells that will build the umbilical cord. And these are the cells that keep on making human choriogonadotropic hormone HCG to stimulate the corpus luteum. Remember, the first trimester in a pregnancy, the embryo must make HCG to stimulate the corpus luteum that will be pumping out progesterone. Without progesterone, the stratum functionalis will shrivel out and will be eliminated from the body through menstruation. So we need this trophoblast for many, many purposes. The inner cell mass will give rise to all organ systems in the body, plus the amnion, which is a kind of protective structure. So at this stage, about 7 days or so into the development, we now have two very different fates for the trophoblast and the inner cell mass. You can see the contact is made. What this blastocyst will do, it will digest its way through the simple columnar epithelium into the connective tissue, and on its way there, the trophoblast can digest glycogen that's built up in the endometrial glands. The embryo is targeting these blood vessels. This is where oxygen and a huge source of nutrients will be coming to it. So when we look at placenta development, we'll keep an eye on how this little embryo can achieve that. So here's the embryo now sunk below the surface of the stratum functionalis. Here's the surface, and this is what it would look like. This is not deep, but close enough to get or to start taking advantage of blood vessels that are in this area. So a few snapshots of the development of the inner cell mass. Second week, 14 days, and this inner cell mass changed tremendously. Take a look at the colors are false colors, obviously, but the shape is not totally different. To my eye, it looks like two hamburger buns. Here's a blue, and here's kind of incomplete yellow hamburger buns. The blue one is called the ectoderm. The yellow one is the endoderm. Here we see yet again cells deciding what they want to become in the future. Cells that are colored blue here will be elements of a nervous system. What the heck does that mean? Well, all parts of the brain. Think about chapter 14, brainstem, diencephalon, cerebrum, cerebellum are all going to be formed by these cells. How about spinal cord, whether gray or white matter, all coming from this, and as well as peripheral nerves. And interestingly, epidermis is also going to be developing from this blue cell layer. The other hamburger bun, the yellow one, this is going to lead to the development of all organ system whose lumen is communicating with the outside world. So what could that be? Well, how about digestive system? How about respiratory system? How about urinary system? Some aspect of the reproductive system are all forming from these yellow colored cells. And then this is the event of gastrulation between the two hamburger buns. A new cell layer start to develop, push these cells apart, push these two hamburger buns apart. This reddish colored mass of cells is called the mesoderm. And a mesoderm is where most connective tissues will come from. Think about bone, cartilage, tendons, most muscle will come from, and also the connective tissue region of our skin, the dermis. So this is 2 weeks into development. So this is, in other words, this is 1 week after implantation. Here is another snapshot. Now we jumped 4 weeks ahead. Neurulation is the term for a more visible development of our nervous system. So if you take a look at here, this blue area is a tube-shaped beginning of our nervous system. You see it here too, and here too. Brain, spinal cord, peripheral nerves, as we said about the ectoderm, this blue-colored tissue. And the heart is starting to form from the mesoderm, and blood vessels are starting to form as well. This is 4 weeks into development. By the 8th week of development, the shape of the embryo is a lot more animal-like. First, it's more like a tadpole shape, and then it slowly turns into the shape of a more recognizable human fetal shape. So now we're talking about the 2nd month, the last month of embryonic development. Brain development occurs. Here is where now the cerebrum, cerebellum, brainstem, diencephalon are all taking shape. Cells are dividing like crazy. Limbs start to grow, and the heart is now 4-chambered. So the complexities of the heart that we saw, not only the atria and ventricles, but connection to the blood vessels, and also valves are starting to form. So we mentioned that after the 8th week of embryonic stage, the fetal stage is just further elaboration of that. So this is where we take a break from the development of the embryo and the fetus itself. And this developing embryo has a tremendous conundrum. Where am I going to get all the oxygen and nutrients? The embryo, as you can see here, the blastocyst developing and implanting here, digesting its way into the uterus, has no food source of its own. The lumen of the uterus is very, very poor in energy carriers and oxygen. So an oxygen-starved and calorie-starved embryo is digging through the stratum functionalis like crazy. So remember chapter 25, we mentioned all these ATP producing processes, glycolysis, coenzyme A formation, Krebs cycle, electron transport chain. Well, these cells have all that operating in them, but they need oxygen, right? Mitochondria would not work without oxygen, and they need those nutrients, glucose especially. So this embryo is in search for all those starting chemicals. The starting chemicals are present in great concentration in the maternal blood vessels, and the embryo is going to figure it out how to gain these nutrients and oxygen in the maternal blood vessels. So one tool in that digging digestion is this kind of yellowish colored set of cells. These are fetal cells. This is the syncytial trophoblast. And what syncytial trophoblast does is that it expands. In some scientific circles, the cells of the syncytial trophoblast are acting very similar to cancer cells. They spread and they digest the stratum functionalis. During that digestion, they digest the walls of the blood vessels, and the maternal blood start to pool into these large cavities inside the syncytial trophoblast. So here's one trick the embryo is beginning to use. Oxygen and nutrients can travel through these tissues by diffusion, and so now the oxygen and blood sugar from maternal blood can diffuse to the embryo and maintain this breakneck pace of cell division and development. So this is the beginning of access to maternal oxygen and glucose. Here's another snapshot. You can see a little bit more elaborate syncytial trophoblast forming these large cavities, and guess what? These large cavities are all now filled with maternal blood. So this embryo is now surrounded by maternal blood, rich in oxygen, rich in glucose, and diffusion is bringing all those starting chemicals to the embryo at 360-degree angle. The embryo itself will be connected to this syncytial trophoblast because in a few snapshots we'll see that this pool of blood connected to maternal blood vessels will turn into the placenta, and this connecting stalk will turn into the umbilical cord. Here is a lot more advanced look. This is not quite ready as a placenta. We are at 21 days. Placenta completes its formation about 3 months, but you can see the idea. Here is yet again maternal blood vessels. The blood is pooling into these large cavities in the syncytial trophoblast, and now we have chorionic villi, these thin protrusions that increase the surface area, and now we have blood vessels of the fetus collecting oxygen and nutrients through the umbilical vein and bringing the waste products from the developing embryo through the umbilical arteries. When the placenta forms, this is the structure that we expect. So we have been talking about chorionic villi. Here is the most elaborate structure of those chorionic villi, many branchings, huge surface area. And these chorionic villi are now completely surrounded by maternal blood. This red-pink substance is maternal blood. Huge amount of oxygen and glucose, now fatty acids, can diffuse into the fetal blood vessels, and all that oxygen and nutrients will be heading towards the fetal body through this red umbilical vein. And waste products, urea, hydrogenized ammonia, are coming to the placenta through the umbilical arteries and being loaded into the maternal blood. Now, maternal blood circulates here. Maternal blood is coming here through the arteries that was built in the stratum functionalis, and deoxygenated blood leaves the placenta through the veins of the stratum functionalis. So this is the crowning achievement, all thanks to progesterone. Remember, progesterone maintained this rich set of blood vessels in the stratum functionalis in the secretory phase. That is maintained. Here we go at the end of the third trimester. Corpus luteum is still churning out progesterone, but that progesterone production is now turned over to the placenta. And the placenta is now fully equipped not only to exchange nutrients and waste products between the maternal blood and the fetal blood, but also to produce progesterone. A really important thing to observe, that there is no direct connection between maternal blood and fetal blood. The two are always separated by a layer of cells representing the chorionic villi. So maternal blood never actually flows into these blood vessels, and fetal blood never flows into the maternal blood. So they are always separated. Chorionic villi is doing that job. Maybe one more clinical aspect here about chorionic villi. Chorionic villi sampling, CVS, is a clinical procedure to check for potential genetic malformation of the fetus. This is done usually when the age of the mother is older than 36, although current guidelines might be a little different. If there's any suspicion of some genetic malformation of the fetus or genetic abnormality, a biopsy can be done taking a chorionic villus, because this is fetal material. Analyzing the DNA from this chorionic villus can give direct information about the genetic makeup of the fetus. So chorionic villi sampling is checking on the genetic makeup of the fetus and potentially identifying genetic abnormalities. So this is what the placenta looks like after delivery. You see the umbilical cord cut, and we mentioned this in the lab that we have the umbilical vein, and I mentioned this on a previous slide too, that carries the oxygenated nutrient-rich blood towards the fetal heart. By anatomical definition, any blood vessel that carries blood to the fetal heart has to be called vein, despite the fact that it's oxygenated blood. And the flip side of it is that the blood vessels that carry deoxygenated blood to the placenta are called umbilical arteries, because they are taking blood away from the fetal heart. And by anatomical definition, blood vessels that carry blood away from the heart, we have to call them arteries. I'm not going to say much about the fetal period. This is mostly elaborating of the already formed organ systems and making them ready for the time this fetus will be delivered. So at the time of delivery, a number of new situations facing this infant. Maybe the most significant is the respiratory system. This fetus for 9 months received oxygen from maternal blood through the chorion aquili, and the umbilical vein carried it to the fetus. The lungs developed, but they were unused. So here comes delivery. The cord is cut, and this means that the umbilical vein carrying oxygen to the fetus is cut too. So what's happening in a fetus? Just after delivery, the fetus is feeling like, or this newborn now, newborn is feeling like you feel when you hold your breath. Carbon dioxide builds up in the blood because no more oxygen source, and also we cannot eliminate carbon dioxide. And if everything develops right, the respiratory center in the medulla oblongata, this medullary rhythmicity center, will be stimulated to cause the first excuse me, first muscular movement of inhalation. So diaphragm contracts, external intercostals of this newborn contract, perhaps the first time functionally, and the infant will be taking his or her first breath. This moment is watched very, very carefully. Obstetricians, neonatologists are there usually, and if this infant is turning blue, it means that the lungs are either not working properly or not working at all, and a quick intervention will remedy this situation. The respiratory rate is 45 inhalations per minute, and after that it is going to normalize to 12 to 15 inhalation per minute. The second little slower adjustment is happening in the cardiovascular system. Way back in the heart chapter, we mentioned two fetal remnants on those heart models in the lab. So here they come again. Foramen ovale. Normally, the foramen ovale is an opening between the left and right atria. So now this is going to close, and the closed foramen is now a fossa. Fossa ovalis is a little depression in the right atrium. Now all the deoxygenated blood will be traveling to the lungs for the first time, so the respiratory system can get its functionality up. Deoxygenated blood of the fetus, of the newborn, will have to be oxygenated in the lungs. So this is an important closure. And the other structure we mentioned on the fetal heart, ductus arteriosus. I'm not going to say anything about umbilical vein and the ligamentum venosum. This would take us deep into the fetal circulation, the subtleties of the fetal circulation. Let's stick with the item that we did mention in the lab. So ductus arteriosus was an open vessel between the pulmonary artery and the aortic arch. This blood vessel, the ductus arteriosus, allowed deoxygenated blood from the pulmonary artery to flow into the aorta. This is no longer desirable, so this blood vessel will shrivel and turn into a ligament. So ligamentum arteriosum is what's left, and here yet again all the deoxygenated blood from the pulmonary artery will be now diverted, flowing at full speed towards the lung, where it will be oxygenated. Here is a similar very high rate of functioning in the cardiovascular system. So at birth, 120, 160 is the pulse. Remember, respiratory rate was up to 45 per minute, and in the weeks following birth, this heart rate will also will get much lower. So this is the end of the last chapter, chapter 29 of the AMP2 semester.